The GPS nanoparticle platform effectively and accurately transports therapeutic cargo specifically to cancer cells. Greetings and salutations to everyone. Today we will delve into an intriguing subject. Penn State researchers have made a remarkable breakthrough in the field of gene editing by creating a GPS nanoparticle. This innovative technology has the ability to transport gene-editing proteins directly to cancer cells, effectively impeding their spread. To ensure successful delivery, the nanoparticle is cleverly disguised with fatty molecules that closely resemble natural lipids. Within its core, it carries CRISPR-Cas9 molecules capable of pinpointing specific genes within a cell's genetic material and either suppressing or neutralizing them. In this study, the researchers focused on targeting the human forkhead box C1, FOXC1, gene, which plays a crucial role in initiating metastasis. What sets this nanoparticle apart is its ability to activate solely within the targeted environment, minimizing the risk of off-target delivery and potential harm to healthy cells. Without further delay, let us commence our discussion on this groundbreaking development. With its unique homing capabilities, the platform becomes active in cancerous environments, where it then deploys gene editing tools. On the 11th of March, their findings were made public in ACS Nano, accompanied by the submission of a provisional patent application for the technology outlined in this research. The ability to locate the precise destination is a feature of the GPS nanoparticle we have created, stated Dipanjan Pan, the esteemed Dorothy Fair Huck and J. Lloyd Huck Chair Professor in Nanomedicine and Professor of Nuclear Engineering and Materials Science and Engineering at Penn State. Upon arrival at the intended location and exclusively at that location, the nanoparticle is capable of administering gene-editing proteins to halt the dissemination of cancer cells. While challenging, our study demonstrated the efficacy of this system in addressing basal-like breast cancers. Basal-like breast cancers, much like triple-negative breast cancers, may not be as common as other types of breast cancers, but they present a significant treatment challenge due to the absence of 3 specific therapeutic targets that are typically found in other breast cancers. Additionally, these cancers display aggressive characteristics, characterized by rapid tumor growth and the release of cells that can migrate to other parts of the body, leading to the formation of secondary tumors through a process known as metastasis. According to Pan, the issue of metastasis poses a significant obstacle, particularly in the case of aggressive breast cancers such as triple-negative and basal-like breast cancers. Detecting these types of cancer can be challenging, as they do not appear on routine mammograms. Additionally, they predominantly impact younger individuals and African Americans who may not have access to preventative care. Unfortunately, the prognosis for these patients is often extremely unfavorable, underscoring the pressing need for more efficient treatment options when the cancer has advanced beyond the early stages. Using a clever approach, the researchers created a nanoparticle that masquerades as a Trojan horse. To achieve this, they cleverly concealed it with custom-made lipid molecules that closely resemble naturally occurring lipids. Furthermore, they loaded the nanoparticle with CRISPR-Cas9 molecules, which possess the ability to pinpoint specific genes within a cell and either suppress their activity or render them useless. In this instance, the focus was on the human forkhead box C1, FOXC1 gene, which plays a role in initiating the spread of cancer cells. The lipids created by Pan are referred to as zwitterionic, which indicates that they possess a shell with a charge that is almost neutral. This characteristic serves as a disguise for the nanoparticle, making it appear harmless and preventing the immune system from attacking it. Additionally, this design allows for the controlled release of the payload, but only in the presence of a low pH environment specific to cancer cells. In order to ensure that the lipids only activate in this acidic tumor microenvironment, the researchers engineered them to transition to a positive charge upon entering this environment, thereby initiating the release of the payload. Given the expansive nature of the body, the challenge for researchers was to guarantee that the CRISPR-Cas9 payload reached its intended destination. In order to achieve this, they employed a strategic approach by incorporating an epithelial cell adhesion molecule, EpCAM, to the nanoparticle, as it is recognized for its ability to specifically bind to basal-like breast cancer cells. According to Pan, there has never been an endeavor to specifically target a basal-like breast cancer cell using a delivery system that responds to the surrounding environment and has the ability to genetically silence the targeted gene. We have achieved a groundbreaking milestone by demonstrating that this feat is indeed possible, Pan declared. There are various methods that have been devised to deliver treatment to cells, including viral delivery systems and non-viral delivery systems that utilize nanoparticles. However, what sets our team's approach apart, according to Pan, is the incorporation of a surface lipid that specifically reacts in the desired environment. This significantly minimizes the risk of off-target delivery and any potential damage to healthy cells. Additionally, Pan noted that because the body does not perceive these lipids as a threat, there is a lower likelihood of triggering an immune response, which was confirmed through their experiments. To validate the efficacy of their approach, the team initially conducted tests using human triple-negative breast cancer cells. These experiments confirmed that the nanoparticle effectively delivered the CRISPR/Cas9 system to the intended environment. In a mouse model, the researchers further demonstrated that the nanoparticle was capable of locating the tumor, activating the system, and successfully suppressing the expression of FOXC1. Following this, Pan mentioned that the researchers have intentions to further examine the nanoparticle platform with the ultimate objective of eventually implementing it in clinical applications for human use. Pan expressed that they are actively investigating additional applications for the platform technology. They are considering the possibility of modifying the surface molecules and payload to promote healing in different regions. The platform holds immense potential for various uses. During the study, Parikshit Moitra, an esteemed research assistant professor of nuclear engineering in Pan's laboratory at Penn State, played a pivotal role as the first author. Currently, Moitra holds the position of an assistant professor at the prestigious Indian Institute of Science Education and Research in Berhampur. Additionally, the research team comprised dedicated individuals including David Skrodsky, Matthew Molinaro, and Nivetha Gunaseelan, all of whom were doctoral students at Penn State. Furthermore, Dinabandhu Saar from the University of Illinois, Urbana Champaign, Teresa Aditya, a postdoctoral researcher in nuclear engineering at Penn State, and Dipendra Dahal and Priyanka Ray, both postdoctoral researchers from Pan's previous institution, the University of Maryland, Baltimore, contributed significantly to the study. Funding for this work was provided by Penn State, the University of Maryland, Baltimore School of Medicine, the Centers for Disease Control and Prevention, the U.S. National Science Foundation, and the U.S. Department of Defense Congressionally Directed Medical Research Program. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.